going to talk about the biomechanics of pregnancy. I was saying before, I am Lullish. It's such a great opportunity for me as a mechanician to present this work in front of a group of mechanical engineers, specifically understanding solid mechanics. On the cover here, I have a shameless plug. This was a couple years ago, but I edited 2 editions of Interface Focus with work on bioengineering and women's health. We really want to bring as many engineers as possible into this field of women's health. There's a lot of great mechanics problems here. There's a complete lack of knowledge of how the reproductive system works in pregnancy. So hopefully I'll garner some interest today. So me as a mechanical engineer and studying pregnancy. So this should be kind of an easy sell for you. Here I have a sagittal view of a 24-week pregnancy. Can you see my pointer? Or no? Can you see my pointer here?
Yes.
Okay, perfect. Now I gotta look at this screen. So this is an MRI of a 24-week patient. You can see we've basically segmented out the uterus is here, the cervix is here, the baby's head is here. The fetus is supported and protected by biologically active soft tissue structures. The cervix, the uterus, and the fetal membrane have these brilliant dual mechanical roles. So you can imagine the 39 or 40 weeks of full pregnancy, they have to be protective. The uterus is made out of muscle. It's made to contract. So how does it stay quiescent through the 40 weeks? And how does it know to contract when it's supposed to? The cervix, the structure down here, it has to remain closed during these 40 weeks, and then it has to dramatically stretch and provide a safe delivery without damaging the tissue, because it will have to elastically recoil after the baby is delivered so the mother doesn't bleed out. And then the fetal membranes, famously in movies, you see that old trope of a woman, riding the subway and she'll say, "My water broke." But braking is something that us as engineers study. So how does the fetal membrane remain intact, and how does it time its fracture? So we believe that biomechanical signals such as stress and stretch trigger tissue remodeling events, contractility, and rupture. So these are things that we naturally study. And of course, the mechanical integrity of these tissues are critical for a successful pregnancy. And on the right, you see, I just have a demonstration, and this is for non-engineers to explain that the uterus undergoes this massive kinematic stretching from the time of 16 weeks, and this is to 24 weeks. And then you can imagine how much it stretches out to 40 weeks. So this is something we'd like to quantify as well, the kinematics of stretching. I'll have 2 stories for you today. I have many stories that I can give, but for this audience, my first talk objective is I want to detail the material behavior and evolution of hormone-mediated cervical remodeling. So what we see close up here, and this is a 1 millimeter bar right here. This is mouse cervix in my mechanical tester. This is a non-pregnant tissue kept intact. It's a ring test. And what I'm doing is I'm pulling these sutures in tension. This has about 1 megapascals in stiffness after the transient time-dependent materials die away. This tissue over here is what we call term tissue. The mouse gestation is 18.5 days. So this cervix is about to deliver. You can appreciate the amount of compliance, about the amount of remodeling that needs to happen. The tensile stiffness here is about 1 kPa. So we have 3 orders of magnitude of remodeling. So we're going to talk about the process of this and how we think about remodeling in the mouse cervix. The second story I want to tell you today is we build these finite element models of pregnancy. We base them on ultrasonic measurements that we can get in the clinic. So this is not your high fidelic model of the uterus and the cervix as one would perhaps derive or segment out from an MRI. That's too expensive in terms of the clinic, also too expensive computationally. So we like to sort of, I tell my students, this isn't a low fidelity model, but some kind of a medium fidelity model. We use these models to generate hypotheses on what might be happening. So I'm going to talk about uncovering structural mechanisms of something called cervical funneling. Cervical funneling is what happens when the fetal membranes fall into the inner canal. Clinicians can see this clinically, and that is a sign of a high-risk pregnancy. So we want to understand the structural mechanisms that happen before funneling. So do we have pinching of the uterus here? Do we have failure of the cervical material? So we're going to talk about how we derive these models from clinical data. And the clinical motivation for our work today is preterm birth. Preterm birth is birth before 37 weeks. It remains a clinical challenge around the world. 1 in 10 babies are born preterm, and the rate of preterm birth is increasing in many countries with reliable data. Prematurity is the leading cause of death in children under 5, and survivors can face lifelong medical challenges. Preterm birth is difficult to diagnose and treat, with 95% of spontaneous preterm birth intractable to current therapies, as they have unidentifiable causes or lack of established treatments. So here, preterm birth means spontaneous preterm birth, meaning those not due to clinical indications like C-section or induction of labor, which accounts for 30% of preterm births. The lack of treatment efficacy points to a lack in understanding of the mechanisms that lead to preterm birth, making it difficult to develop effective diagnosis and therapies. The challenge in diagnosis and treatment is particularly highlighted by the fact that preterm birth etiologies, cause or manner of causation of disease or condition, have inconsistent definitions and are still evolving. So historically, preterm birth was split into 2 categories: preterm labor, defined as regular contractions accompanied by cervical change less than 37 weeks, or PPROM, the premature rupture of the membranes, defined as spontaneous rupture of these membranes before 37 weeks. Although preterm birth involves multiple and overlapping phenotypes, we focus on the mechanical etiologies such as cervical insufficiency, the preterm rupture of the membranes, and uterine overdistension. So I want to make it a point here that although being a mechanical engineer, we can study the mechanics of it, I want to make it important that there are many different risk factors that cause preterm birth, and we're starting to learn more and more about them. It's critically important that I work with clinicians and epidemiologists to understand the different factors that cause preterm birth. My work started in the cervix long ago because the final common pathway for many of these preterm birth etiologies is cervical dilation, which results in preterm birth. Also, the cervix is a window into the health of the pregnancy because during a clinical exam, the physician often, you can see the cervix in a pelvic exam, and that's an indicator of the health of the pregnancy. So my overall engineering goal is that, working with people who are trying to understand the genomics, proteomics, transcriptomics, bioinformatics, etc., how can we connect this omics data with biomechanics or perhaps mechanobiology data? And our hope is that we can use computation to have a forward predicting model to identify perhaps clinical biomarkers that are going to tell us that the tissue is going to mechanically overload somehow, so that you have too much load too soon in your pregnancy. So that's my hope with computational mechanics in pregnancy. Perhaps, again, one way, can we get a personalized model? Or again, can we use our computational model to generate what if scenarios to better stratify the risk for preterm birth? And then our hope is that we can feed this risk stratification to the clinician early on in pregnancy so we can get a clear diagnosis. Should we treat this preterm birth? Should we treat it now? And what kind of perhaps mechanical support can we provide? And this is just an ideation device that my students and I were drawing up based on, okay, if we had this type of mechanical overloading, what kind of mechanical device can alleviate that load? So this is where we're headed with our work. We really want to translate these ideas of the cervix using mechanics to build devices that actually work for the patient. Equally important is we want to identify people who perhaps don't need clinical treatment. Again, perhaps the patient has a cervix that's made out of steel. I like to say to the physicians that it's really strong intrinsically, that the biochemical makeup or the material is not going to remodel. Even though the cervix looks short in a clinical exam, you don't need to treat this woman, because overtreating women is equally detrimental, most likely to their health and also clinical costs as well. And so that brings me to my lab. So my lab might look like other labs. It's a traditional solid mechanics lab. We use the tools of theoretical, experimental, and computational mechanics to understand what are the material properties of the various reproductive tissues in non-pregnancy and pregnancy. So we are tasked with formulating microstructurally inspired constitutive models. We work in the cervix, we're working in the uterus and the fetal membranes. We'd like to discern the mechanical role of the biological components that make up these materials, mainly the collagen and elastic fibers and smooth muscle cells and glycosaminoglycans. We measure in many different models. I'm going to show you the mouse and the human pregnancy. We also work in a macaque model of pregnancy, which I'm not showing you today. So it's critically important to use animal models in pregnancy because understandably, it's difficult to study pregnant women. We have an interest in hormone-mediated growth and remodeling, in differentiating hormone-mediated growth and remodeling from inflammation-mediated growth and remodeling. They get two very different materials where hormone-mediated remodeling, you're left with this very sort of strong, compliant material that doesn't want to break, where on the other hand, we found that inflammation-mediated growth and remodeling leaves the tissue embrittled and likes to break. And then I'll show you the finite element framework that we're working on. So I'll pause here for any questions, and we're going to start diving into the work. So hopefully I've inspired some people to work in pregnancy. This is a fun topic. There's not many of us that do this. Okay, cool. It's like I'm podcasting. It's kind of just like speaking into the either world. So, okay, let's talk about the material property evolution of the cervix. So here's a tension curve. These are curves that I published when I was at MIT, a snapshot of uniaxial tension compression of the human cervix. These are non-pregnant specimens from hysterectomies. So these tissues were being thrown away. I biked over to the hospital, picked them up, and then put them in the machine. You can notice here, this is the equilibrium tensile response of non-pregnant tissue in the blue here, in pregnant tissue in the green here. Again, you can see this drastic remodeling that happens between pregnancy. Here, I used a pretty simplistic fiber composite model to model these tissues, where I had an additive contribution of a compressible neo-Hookean ground substance, and we had some kind of collagen fiber model with some type of directionality that we were able to get through some imaging. But really, the take-home message here is that we have lots of softening and tension, but also with any fiber composite material, we have that asymmetry between tension and compression that's quite important for hydrated, soft biological tissues. And this is something that we capture with our model, and that's quite important in physiologic loading of pregnancy. But coming to Columbia, I've been here 11 years now. I really wanted information about the material properties between this blue and this green curve here. So I connected with my colleague Mala Mahendru at the University of Texas Southwestern Medical Center in Dallas, and she's the biological expert on cervical remodeling in the mouse. Here's the mouse reproductive tract. So we have the cervix coming here. We have these interesting two uterine horns that come together in a single cervix. And so if you see a cross section, oh, do I have a cross section of it? No, I don't. What she has published is that the mouse cervix actually softens early on in pregnancy of its 18.5-day gestation. So she has she has quantified the biological changes that happen in the cervix from non-pregnancy out to day 19. And she published these images of the collagen network. So this is second harmonic generation of the mouse cervix in non-pregnancy all the way out to day 18. And then here we have Mason's trichrome stain. You can see that the cervical stroma is dense with collagen fibers, and it's also laced with elastic fibers and glycosaminoglycans. Again, what you can appreciate here as mechanicians is that we have this increase in collagen disorganization. We found that chemically that these collagen fibers, as they remodel, they're less cross-linked. I don't have a slide that says this, but we just published. We know that the cervix actually constantly remodels itself. So the cervix does not get soft because we have less collagen there. Actually, we actually have the same amount of collagen between non-pregnancy and day 18. So actually, if we look at the the biological length scales of the collagen from the nanoscale all the way up to the microscale, what we found, and if you pay attention to the bottom row here, this is the collagen composition in non-pregnancy, and this is it in pregnancy. What we find is we have the disruption of the trivalent cross-linking of the collagen fibers, where the non-pregnant tissue has these very mature collagen fibers as opposed to these immature collagen fibers that disrupt the collagen fiber and make it more disorganized. So for the molecule level study, we could see that it remodels a lot, and this remodeling leads to collagen fibers becoming looser and more disordered structure, and it becomes larger in radius in a TEM image and more wavy as it appears in the second harmonic generation imaging. We've also used optical coherence tomography imaging to look at the collagen fiber bundles, to look at the architecture and the anisotropy of these fibers. The cervix here, this is the inner canal, it's kind of coming right out at you. We found that the collagen fibers ring around that inner canal and have these gorgeous circumferential hoop fibers. These hoop fibers actually stay in pregnancy, but what happens is the collagen fibers themselves are more disorganized. And then this builds up into the cervical structure, and this is an ultrasonic image of the entire cervix in a patient. And I'll tell you more about that in a minute. So this is what we're looking at and what we've measured with MALA over the years. We have a really good grasp of what's happening with the collagen. So now comes, okay, so how do we model this? And how can we mechanically test the mouse cervix at different gestation time points to understand how these changes in the microstructure contribute to the macroscale continuum level changes of the material? So here's my classic representative volume element. So inspired by our fibrous extracellular component, we have modeled the tissue as a porous hyperelastic fiber composite. Again, my total stress is an additive contribution of an interstitial fluid pressure driven by some osmotic pressure of the glycosaminoglycans. And these are things that we can assign. I know how many glycosaminoglycans are there. I know they're fixed charged. And that's added to the hyperelastic solid stress. And we've played with many different flavors of these models. Here I'm showing a very phenomenological solid model, again with a compressible neo-Hookean ground substance and a continuously distributed fiber network. And then the fibers is phenomenologically prescribed here with a simple power law. We've since moved to Langevin chain statistics a la Mary Boyce and her work long ago in polymeric mechanics. So we've interplayed with how serious we want to be with our constitutive models depending on the scientific question that we're asking. So we have many different flavors of this. But what we found most important in these constitutive models is first that you have this contribution of this collagen fiber network, that it is able to, the chains are able to rotate and stretch in the direction of principal stretch. Another salient feature that these models must have is this fact that here's a heavy side step function, is that these fibers only hold tension and not compression. And again, this is something that's classic. I'm not inventing anything new here. I'm just bringing in fiber mechanics, if you will, into the study of the cervix. But so we play around with all different types of models. At some point, the mouse tissue likes to swell a lot. The human tissue doesn't swell as much. So sometimes we get rid of this interstitial fluid pressure. But when we want to study poroelastic effects, we put it back in. So I'm happy to have more conversations about our modeling strategy. But moving forward, we use this rather simplistic model to describe the material property changes of the mouse cervix. Here's an image of the mouse cervix in non-pregnancy. This is a close-up image of it. We like to call it a thick-walled cylinder. My students get mad at me for doing that because they actually handle the tissue. It's much more complex than a thick-walled cylinder, but that's kind of an easy way to describe it. You can see here, this is 18 days. We have the pups here lining the uterine wall, and then the cervix down here, and you can see that the tissue has increased in volume. That volume is dry mass and also hydration as well. And so this plots it here. So if we want to look at the tissue dry mass and volume change throughout pregnancy, there are distinct regimes of losing and gaining density. And let me lead you through this sort of dense slide that I have here. On the left-hand side, this is the hydration level here in these X's. So we do have a slight increase in hydration. But more importantly, what we have is we have a lot of dry weight and wet weight coming on. And so this is the cervix bulking itself up at the end of pregnancy because it has to deliver and it has to go under enormous amounts of stretch to go around the fetal head. And then here are relative changes in tissue volume. And then what I did is I graphed the solid apparent mass density according to the referential and current volumes. And here's a snapshot of the mechanical test. This slide kind of says it all for the material properties. Here's an example of our ring test as I'm pulling in tension. I have a set of 2 DIC cameras so I can capture the deformation in 2 perpendicular planes. We've since used deep learning algorithms to segment out the shape here so we can have we could calculate Cauchy stress at each point. So if I do a ramp pull test, you notice that my non-pregnant tissue doesn't stretch too much, and then it breaks. And because I'm not stretching too much, I'm not activating much viscoelasticity. So it's stiff, doesn't like to stretch too much, and it breaks. Now, as the tissue grows and remodels, what you notice here is I'm able to extend it or stretch it to enormous amounts of stretch. It's very compliant. Its stiffness is dropping orders of magnitude. And with this deformation we have here, you could see that the tissue now is accommodating much more stress relaxation. We believe and hypothesize that this viscoelasticity or stress relaxation is a protective mechanism of the cervix. So when a woman is in labor, she'll have forceful contractions and then there's this pause. You know, it's not this full contraction all the time. So this pause most likely allows the tissue to relieve the stress that's within it so it doesn't break or damage. So this is something fascinating that we're trying to fit a nonlinear viscoelastic model to in tension. These curves here are most likely dominated by the collagen fibers themselves because we're pulling to extreme amounts of tension. And then we just went through the exercise using an inverse finite element analysis to fit our material model that I showed you a few slides ago to the tension data here. And then what we did is we did a model validation where we were looking at our deformation information to see if we were able to capture the unique deformation that happens, the sort of the Poisson's effect of the tissue. And you get this really fun sort of classic fiber composite response in those directions, and we were able to capture that quite well. So we did that with a whole bunch of specimens. And let me back up a minute here. Oh, right here. Okay, so here's our equilibrium material response. This blue line here shows the collagen fiber stiffness, if you will. And then this is the Young's modulus of the ground substance. You could see again the orders of magnitude drop that we're getting with gestation time. This drop in stiffness we do attribute to that disruption of the collagen fiber itself, where the fibers are becoming more immature and then they're losing the maturity. And we have confirmed and validated this measurement with a recent proteomics paper that my colleague Mala Mahendra put out. So just in summary, these are actually the curves that I just showed you for our test. We wanted to compare our mechanical test to mechanical tests that were also in the literature. Most often in this field, the mechanical tests of the cervix, they don't do ramp hold tests. They just measure the sort of instantaneous material response of the tissue. And so this is sort of the tangent moduli of the instantaneous response of the tissue. This is the equilibrium response. Again, we're seeing this viscoelasticity here happening as gestation progresses. We know chemically that at the end of pregnancy, there's a lot of hyaluronic acid at the end of pregnancy. So we are currently hypothesizing that the hyaluronic acid contributes to the viscoelasticity or perhaps the protection of the collagen fibers themselves at the end of pregnancy. So that's work that we're currently doing. So I have nothing to show in that respect. But this was a nice hypothesis generating work between me and my colleague Mala Mahendra. So just to summarize this first part, and then I'll talk about our finite element models. Sort of the take-home point of these animal models. First, we had to develop reliable material testing protocols for the mouse cervix. This took us many years. It's not a traditional mechanical test by any means. The tissue is soft. It's viscoelastic, it's poroelastic. The deformation in 3 dimensions is nonlinear. So it took us a while to figure out how best to interrogate the material properties here. But our take-home message is that indeed the cervix remodels in this regimen of what we call steroid hormone changes. Here's an image of how the hormones change in pregnancy, and this is a graph for the mouse. This is something Mala put together. In pregnancy, we have the steroid hormone called progesterone that's on all the time, and then we have very low levels of estrogen. At some point, these levels of progesterone and estrogen actually flip-flop. So currently we're hypothesizing that these hormones, and we know estrogen actually controls the enzyme that cross-links the collagen, we believe that these hormones are triggers for growth and remodeling. And we know this is evidence here. This is a mouse cervix from a mouse that we took out the ovary, so there's no hormones. And so we're in the middle of doing experiments where we're dosing in estrogen and progesterone to understand how that actually grows the material. So this is a mouse cervix with no with no ovaries. So these are called oviruptorized mice. And then we're adding in doses of estrogen to understand how the material is laid on, and if the new material that is created by the estrogen has the same material properties as one would find in an in situ tissue. So in this regime of high progesterone and low E, we have a loss of solid dry mass. My collagen fiber network, it's priming itself with the loss of cross links due to the low estrogen. And then we have a decline of equilibrium tensile stiffness properties. In the regime with high P, where estrogen starts to increase, I see a continued decline in tensile stiffness. At day 15, I'm at the lowest point of my collagen crosslinks, and I'm transitioning to a glycosaminoglycan regime where I see an increase in swelling and viscoelastic response in my tissue. And then right at the end of pregnancy at day 18, we have a massive progesterone drop. Estrogen is at the highest amount it possibly can be. So we see a major increase in dry mass of the tissue, preparing the tissue for dilation and repair. And then we see immature solid dry mass components do not contribute that much to the tensile stiffness. So that is sort of my take home. I'm going to, again, pause for questions, take a sip of water. Okay, so let's.
Kristin, can I ask a question?
Yeah, please.
So, yeah, very interesting. I have to say that when my wife got pregnant the first time, I was looking for some, I was quite surprised to see at how the knowledge we have is very empirical and not really, I was looking for more mechanistic understanding that I could find. So I got me a little bit confused, but it's okay. Thank you for working on that. And okay, very interesting. One very naive question is, I find this very surprising, this decrease of stiffness, because the cervix is supposed to stay kind of closed until it opens, right? So it needs it still needs to show some stiffness, some rigidity, right?
Yes. Yeah. And actually, so this mouse work that Mala has done, where she has shown in an animal model that the cervix starts to soften right away, early in pregnancy. And a lot of the doctors I talk with, they're like, we know that the cervix softens, but I can't resolve that mechanics question, but it needs to stay closed. And we know it stay closed until 37 weeks. So what happens?
Yeah.
So and I think the next section of my talk will explain why, is because we have the fetal membranes that are holding up the amniotic sac and it's stuck onto the uterine wall. So as the fetal membranes in the uterine wall are taking on the load of pregnancy, the cervix is remodeling underneath it. So if the fetal membranes start to lose its mechanical function and the adhesion between the fetal membranes and the uterine wall, if that starts to lose its mechanical function, and if your cervix is too soft, then you're in trouble.
Okay.
So we believe, we hypothesize that there's a safe way for the cervix to remodel, and that it does have to remodel because it does have to prepare itself at 40 weeks to undergo stretching of, north of 2, stretch ratios of 2, 3. What type of material can undergo that type of stretch?
But there is, like the material gets prepared for it, but there is a structural effect that maintain it closed.
Yeah. But I do hypothesize that women who truly have a very soft cervix, what happens is they start to funnel. And that actually beautifully leads me into my next section of my talk, is now let's look at the structural mechanics of exactly what you said. So, and thanks for that question. That was like, it's like I planted it. So how do we uncover these structural mechanisms of clinically observed funneling? So this is where I want to translate the mechanics work, and where we sit at the bedside with the patients and with the parents to try to understand these questions. This is a transvaginal ultrasound of a woman's cervix. So the ultrasound probe is here. It's inserted into the vaginal canal. It sits up against the cervix. The cervix is the structure I'm outlining here. Here's the fetal head. This woman has a nice long cervix. It's closed. You could see a lot of soft tissue material here. This woman has something what we call funneling, and this woman would be placed at high risk. So what happens is the fetal membrane is falling into the inner canal. And I have a cartoon of this. So in a normal pregnancy, you have the cervix, the uterus, and the membranes. As pregnancy progresses, and this is for a healthy pregnant, you have effacement of the baby. So maybe about a week or two, the baby starts to descend into the pelvis and it lays off the diaphragm and you can talk again. And then when you get to this point, you're ready to be admitted into the hospital, and then the doctor watches the dilation, 1 centimeter, 2 centimeters, 3 centimeters. For some women, this process goes awry and we're left with cervical funneling. So what happens is the fetal membranes fall into the inner canal. And they can see this in the ultrasound, like I showed you. And then this is a funnel where it goes into a V and a U shape. So this funnel can happen for 2 reasons. 1, the cervix is too soft too soon, or 2, the fetal membranes have somehow become undetached from the uterine wall and they fall, and there's just too much load on the cervix. I believe it's a combination of both. I believe that 2 mechanisms are happening and they're probably feeding each other. I believe that the cervix for women who deliver preterm is truly too soft and too soon, increasing the shear stress of that adhesion between the fetal membranes. And then it starts to become undone. And then now it's applying more weight on the cervix. And then the cervix keeps opening and opening and opening. So it's this circular effect. And I've been able to hypothesize exactly what I just told you because I've built these finite element models to look at exactly that. So let's look how we build those finite element models. So again, clinically, standard of care in the United States, and at our hospital at least, every woman between 16 and 24 weeks gets a transvaginal ultrasound. They measure the length of the cervix and they say, okay, if your cervix is below 25 millimeters, you're in trouble. So the physicians have a mechanical intuition that cervical structure is important. They measure cervical structure, they measure the length. It baffles me that they don't measure the diameter as well, but I'm trying to get them to do that. But this is it. But I often tell them if the short cervix is really strong, then it's not going to open. And I keep telling them if this long cervix is very soft, it could be at risk of opening. So obviously there more needs to be done. So making computational models in pregnancy is tricky. Pregnancy obviously is a protected environment. And you want to be careful with the research protocols that you ask a pregnant patient to do. And I'll say that my research protocols have drastically changed after I had my daughter 6 years ago. Well, before I had my daughter, I was making these really high fidelity pregnancy models based on MRI images. It was a lot of money. It was a lot of time. And I was only studying maybe 2 or 3 women at a time at a singular gestation time point. So what I told myself was, and I made this conscious decision, I said, I want to study way more women than just 5. I want to study all women. And the only way to do that is if I build parametric models of pregnant anatomy and to capture the most salient features of the structure. And that's exactly what we did. So we started working with the sonographer, the clinicians, the patients to design better research protocols to study women. I think this was something that was really important that sort of broke open the doors of what I do. So this is the ultrasound protocol that we came up with. This is really easy. It takes, I don't know, 15 minutes with a trained sonographer. So what we do is we take these sonography probe, we could drag it across the belly. We can get these overall dimensions of the uterus. We can get the thickness of the uterus. We can get the placement of the uterine axis with the cervical axis. We can sweep it again left and right on the belly. We can get these overall dimensions left to right. And then we can take a transvaginal scan, which again is standard of care in the United States, where I can get these very detailed geometric measurements of the patient. As you can imagine, all of these structural elements are going to play into how the load is set up on the cervix, how the uterine wall is pulling the cervix open, etc. But now I've developed a really sort of easy way to study a lot of women. And now we're building these large cohorts of clinical measurements that we're releasing to the world so anybody can study this. Another important factor, I wanted to measure cervical stiffness. So how do you do that in vivo? This is actually a project that Eduardo Mazzo at ETH Zurich had started in his laboratory with Sabrina Bader. She was a graduate student when I met her. She's now the CEO of Pregnolia, and she has this beautiful device. We have two of them at our medical center. It's a pump, it's an aspiration tool. I don't have to explain to the mechanicians how that works, but we apply a speculum during pregnancy, and we can put this at the end of the cervix and suck up 4 millimeters and measure closure pressure. And then I can use some inverse finite element way to get at some kind of stiffness measurement. And that's exactly what we do. I love this device because it's not scary. My clinicians just have it in their purse. They can go to their clinic, measure the cervix. This takes 3 minutes. But this measurement gives us a lot. So with these research methods, I have involved many clinicians at the medical center. And so I'm in the middle of collecting clinical data for a low risk cohort I'm going to call Lori. We see them at 4 time points in their pregnancy. We're recruiting 50 women. At each research visit, we do a ultrasound scan for the maternal anatomy. We take a cervical stiffness measurement, and then we also take different swabs of their cervical vaginal fluid to work with somebody in microbiome and do sort of an omics analysis as well. And of course, we collect maternal data from their electronic medical record. And actually, we've been doing this for the woman standing up and laying down. We actually found no difference in the maternal anatomy between standing up and laying down, and because of COVID protocols, we wanted to reduce the research time. So we only do measurements with the woman laying down. And then we also have a cohort of high-risk women. These are women who are getting a biomedical device called a pessary. The results I'm going to show you today are from the non-intervention arm of this clinical trial. I can't talk about the pessary today because it's still in clinical trial. So these patients are identified with a short cervix between 16 and 23 weeks of their pregnancy. So we take their maternal anatomy and cervical stiffness at the time of pessary placement, or sometimes, and then again, some patients don't get it. And then we also do a follow-up visit here. And so I'll show you 12 models from them. Here's our parametric model. We built this in SolidWorks. This parametric model. The reason we did that is because I train a lot of undergraduate students, and I want to get more engineers excited about pregnancy and understanding maternal anatomy, and this was a great tool to do it. I used to code, I used to use all these sort of like proprietary softwares and codes to build these Boolean models, but SolidWorks is fine. And I am happy to share the SolidWorks file with anybody who wants to play around with it. So we're able to generate these models pretty quickly. I'm going to fly through this, and I can talk about the details, in the Q&A. Currently, so for every patient, we build their solid model. We mesh it. We've been playing around with different element types. We use hypermesh. You can see the different element types. We're working through mesh conversion studies at the moment. Our material properties, we assign based on existing tensile data and gestationally determined cervical stiffness, but also patient-specific cervical stiffness. I can go into the details of all of these material properties. All of them are nonlinear. The fetal membranes, the uterus, and the cervix are all fiber composite models based on published mechanical testing data sets. A lot of the fetal membrane is from Eduardo's lab at ETH. We have uterine data now, and I have cervix data of my own. And again, we just do traditional material model fits and I can go into depth about this. These are our boundary conditions. You can see where we have tide and sliding. Currently for healthy membranes, we tie it, and then we have some sliding here. This tied boundary right here mimics the pelvic floor that's holding up the uterus into the abdominal space. We work with a pelvic floor, Steve Abramovich at Pitt, to sort of like make these low fidelic ways that we can capture something that's very complex. So we're still working on that, but we're quite happy with sort of this low fidelic way to capture these things. And then of course, these are some boundary conditions. And lastly, we assign a load, which is a gestationally determined intrauterine pressure, accounting for intermembrane, which we apply to the inner membrane surface. And this completes our FEA setup. And so this paper here has actually measured the amniotic pressure at various gestational time points. So let's look at data. So, and I also want to say we make these models so in the 16 to 24 week gestational period. And when we create these models, we make these in a reference configuration for the cervical length. A lot of our patients already have a funneled cervix, but we tell the model to start the simulation with the cervix closed, and then we're going to model cervical funneling for our patients. Okay, so let's look at the stiffness of the cervical stiffness of the cohort of patients. So the blue circles here are our high risk patients. On the y-axis here, we have closure pressure. As you can imagine, the higher the closure pressure, the stiffer the tissue. The lower the closure pressure, the softer the tissue. In the blue are high risk patients. In the gray are our low risk patients. You can notice too, the softest of cervices for two patients, again, take this as hypothesis generating. These women delivered extremely preterm. Notice up here that I have a mix of blue and gray. So our low risk patients had a, kind of a large range of, closure pressure is on the x-axis and gestational age is on the y, pardon me. You can notice that our low risk patients had a wide varying of cervical stiffnesses, but they kind of delivered close to term. And also a lot of my high risk patients delivered around term, but they had these increase in funnels. So this really goes to show that these intrinsic material stiffness of the cervix does matter. All right, so let's put this all together for all of our patients. So here's a typical simulation where I'm showing the first principle Lagrange strain in the uterus and the cervix as the intrauterine pressure is applied. You'll notice as the fetal membrane pushes into the cervical canal, the internal os is compressed as well as the uteral cervical junction and the lower uterine segment. So here's the inner canal and here's the cervical uterine junction right here. And let's look at it. Let's look at everybody more closely. So to visualize what's happening in the proximal end of the cervix where the internal os and uteral cervical boundary are located, we mapped out first principle strain and the strain direction being noted by the black lines. All of the circumferential strain at the internal os, and as one moves out, so we have circumferential strain here, and then as one moves out, you see all of that radial strain and stress from the uterine wall tension. And then if we just look at the strain and then plot it with gestational age, of course, the women with the softest of cervix that are experiencing the most strain end up delivering early. And then this is kind of the punchline. This is the end of my talk. This is a graph that I've been working on for the last 11 years of my career. So if we group the results into cohorts, there's a clear difference between the high and the low risk groups. There's a distinct pattern of cervical strain and stress that is associated with a short cervix and cervical funneling. Overall, we see greater magnitude and strains in the high risk group, with the highest strains happening at the uterocervical junction. In a different study, we show the pattern of strain are driven by overall uterine shape. Of note, again, in these cohorts, we had two patients with a very soft cervix that delivered extremely preterm. So this patient here and this patient here were the patients with the softest of cervices. So again, we modeled the cervixes as they were closed. And what was happening in the mechanical environment for these two patients is what was driving these huge strains is first, the cervix was too soft, but also it was experiencing a lot of uterine wall tension. So that leads to the hypothesis is that the thickness of the lower uterine wall might be a key structural factor to look at clinically to understand how much load is on the cervix. And so just looking at results more closely, a high magnitude radial strain at the uterocervical junction is associated with clinically observed funneling. Here you can see the strain maps of the proximal cervix with the mask of the patient's transvaginal ultrasound. So if we just masked out the cervix and the uterus from the actual clinical image, you can see it better. And you can again see that this extreme funneling that we see is associated with this type of mechanical environment. So huge strains at the uterocervical junction. So key mechanical factors, the uterus, fetal membranes, and the cervix all share the load. Membrane adhesion and stiffness are critical factors. I talked about the material qualities that one must have in these models. Also, I didn't go into it too much, but maternal anatomy is extremely diverse. Maternal structural factors can exaggerate the loading for the cervix. So if you have an exaggerated load on the cervix in combination with a really intrinsically soft cervix, then you should be placed at high risk for preterm birth and the correct clinical action should be taken. So with my work, again, I'm hoping that we have new structural biomarkers that can indicate if a woman is going to be at risk for delivering preterm. And this more of the same, a lot of limitations. You could see in that model, I could go through every single limitation I have in model improvements. They keep coming. We've got some new big steps. We're doing some fun AI deep learning work to automatically segment these medical images and combo that with the electronic medical record data. We're stiffness mapping the human uterus for the first time. This is OCT images of the smooth muscle cell fascicles. So we've measured the anisotropic material properties of the human uterus. That has a lot of implications in implantation mechanobiology and gynecologic disease. There's a lot here. There's only one of me. I just wish I could share this work and invite people to work in this space with me. And I do that by open sourcing all of my work. You can find it we have this website, SimTK, that I've been working on with Steve Abramovich, but I also put everything in my library at Columbia that you can download. You can also email me. So I'm just going to end there, and I like to thank my team. So thank you for having me.